you mentioned before, when you reflect on your first experiences, your personal start and beginnings as a student or even early physician, can you see and maybe outline for young listeners that whether there were important events or even personnel mentors that influenced you?
Sure. So actually, it fits quite nicely. So for me, when I was in my last year of medical school in 1979, I read a paper in the New England Journal of Medicine that came from Seattle, from the transplant program, on the graft versus leukemia effect. And that was what really, at the time, I, was very much interested in hematology, and I'd been, seeing leukemia patients and seeing the treatments they undergo with chemotherapy, and it was sort of my first introduction to transplant, but also an introduction to this idea that the immune system was involved in eliminating the leukemia. And my second interest, as scientifically, was in immunology. So it was at that point that I really decided that my future, I wanted to go to Seattle and train in transplant. I was fortunate to go there in 1985 when Dr. Thomas was still active. And it was extraordinary to actually work in the transplant unit in those early days. What became clear to me as a fellow on the transplant unit was that most of the major problems that we encountered in transplant were immunological. So there was this enigma called the graft versus leukemia effect, but it lacked specificity. We had no way of really separating it from graft versus host disease. And then there was, of course, the opportunistic infections that we observed, again, due to an immunodeficiency. And so much of my early work really focused on trying to understand what were the immunologic mechanisms, and everything honed in on T cells. So I became very interested in T cells and in the idea of being able to treat patients with T cells that had exquisite specificity. So it was also in 1985 that Alan Townsend and Andrew McMichael published a paper in Cell that demonstrated that CD8 T cells actually saw a peptide presented by the class 1 MHC molecule. This was shown in influenza, but it was now a demonstration of the exquisite specificity of a T cell, that it would be specific for just that single epitope of a virus, for example. And it struck all of us at the time that, well, if we can find these targets on tumor cells, on cancer cells, why couldn't we actually use T cell therapy to treat cancer? So I started out, though, my early work in T cell therapy focused on an easier problem, and that what I thought was an easier problem, and that was to reconstitute immunity to a virus. Because we now knew that T cells see viruses and they see these viral epitopes, it seemed to me that potentially we could reconstitute immunity, for example, to cytomegalovirus after allogeneic transplant. And indeed, what we did was we embarked on an effort to do that. Now, this did not involve genetic modification at that time. This was the late 1980s, early 1990s. The ability to easily genetically modify cells was just not possible. So what we did is we isolated T cells that were specific for CMV from the bone marrow donor, and then used them after transplant to reconstitute the patient. And that was really the first efforts. This was done with Phil Greenberg, who I studied with at the time. It was really the first efforts to reconstitute antigen-specific immunity in humans. And as you probably know, efforts to reconstitute CMV immunity and EBV immunity is still going on after these many years. But we had success in doing that. We could demonstrate we could transfer immunity. We could find the cells that we transferred into the patient, that they persisted potentially for many months, we demonstrated, but potentially even longer than that, and that they had an effect against the virus. So then, of course, I'm an oncologist, a hematologist. I wanted to treat cancers, not viruses, even though it was a very exciting work, and gradually moved my lab towards this idea of being able to specifically target cancer cell antigens. And finding T cell receptors or T cells that actually saw leukemia was actually not so easy. And I had several postdoctoral fellows come through my lab that worked on this. Many are now on faculty at various places, some in Seattle, some in Japan and around the world. And they continue to hunt for these antigens that we can target on leukemia cells. And they found some, and we've actually been in the clinic with that approach. But it is really, really challenging. And so, of course, at this time, we also started to realize that the advances in other areas, particularly the ability to use retroviral vectors or lentiviral vectors to introduce genes into T cells. And that made it now possible to start to imagine to introduce receptors that would target cancer cells. Now, the CAR field really started almost peripheral to all of this work. It was Zelig Eshar who was interested in making receptors that would retarget a T cell to a cancer cell. And he did this work in the late 1980s, early 1990s, where he linked the binding domain of an antibody, so what's called a single chain variable fragment, which encompasses the heavy and the light chain of the antibody, that would target a molecule on the surface of a cell, now not an MHC peptide complex, but actually any surface molecule. And he linked that to T cell receptor or other immunoreceptor signaling motifs. Rick Klausner had discovered the critical role of the CD3 zeta chain in transmitting signaling in T cells. And so Zelig, very quickly used that information to link an scFv to a zeta chain molecule and demonstrated that these would signal. So you could put them into a T cell and they would induce a signal and activate the cell. Of course, it took many years for people to really understand the design, and indeed this work is still going on, to understand how do we optimally design a receptor. I think the next advance really came from several groups, Dario Campagna at St. Jude, Michelle Sadelaine at Memorial Sloan Kettering, and Michael Jensen at City of Hope, demonstrating that if you added a co-stimulatory molecule to the zeta chain, that you could actually invoke co-stimulation. But I would point out that all of this was done empirically. It's sort of say, well, we know a T cell gets co-stimulation, so let's put a co-stimulatory domain on. And it wasn't really done in an informed, other than empirical information, but done in a way where we actually studied what were the consequences of introducing those domains into the chimeric receptor, and how did that affect cell fate and function. And it's actually only now, even after we got to the clinic, it's only now that we're really starting to dive into that more rigorous science, I think, that will lead to a next generation of receptors. But these receptors that were developed both in all of these labs, including in my lab at the time, were highly effective in eliminating leukemias or lymphomas in mouse models. And what we did in those models was we would take mice, there's strains of mice that completely lack an immune system. So these are called NOD/SCID gamma receptor knockout mice. They have normal mouse hematopoiesis, but they do not develop T cells, B cells, or NK cells. And so the advantage of those mice is that you can now engraft human cells and tissues and tumors into those mice, and they will grow just like they would grow in a patient. But of course, if you engraft a human lymphoma or leukemia, you can also treat it now with these T cells that are engineered to have these receptors. And so we could demonstrate in those models that these T cells were highly potent in eliminating these tumors. And I think that really provided encouragement, gee, maybe they could work in patients. So that was a very, I think, exciting time. It gave us models where we could study not just the receptors themselves, but what kinds of T cells should we put the receptors into, for example, and work in my lab by actually two German students that were in the lab at the time, Michael Hudacek and Daniel Sommermeyer, demonstrated that particular subsets of human T cells were more potent in eliminating the tumor. And this is actually starting to be borne out now in the clinics many years later from their initial work. And I think I'll come to that when I talk about, I think, where the future of this could go. So this was all a very exciting time. I mean, we thought we had the tools to move this to the clinic, but of course, there are great technical challenges from treating a small mouse to moving this up to patients.
When was that? And
so this all was occurring in the late 1990s and the early 2000s. I think almost all the groups, by 2005, 2010, really felt that this needed to go to the clinic. But I will tell you, and you can, Carl June will allude to this, is that when we put in grants to the NIH to move this stuff to the clinic, they were not favorably reviewed. And so,
Why?
Part of it, well, it's interesting. I think part of it was the funding in the United States often is very tight. And I think part of it was people just weren't convinced that this was ready, that we really understood enough about these receptors, that we understood. It's one thing to be able to treat cancers in mice. I've often said that, we cured cancer in mice in the 60s and 70s, and we're still having trouble in patients. So I think there was a bit of skepticism just about the viability of this approach. The complexity, remember, you're genetically modifying cells. You have to grow these cells up ex vivo before you put them into patients. And of course, we're putting in these receptors that are synthetic. They're man-made. They're not natural. So there was a lot of skepticism, I think, that this wouldn't work, or that it would prove to be too challenging. There were some funding agencies that did see the potential of this. Leukemia and Lymphoma Society, for example, I think funded some of Carl's early work, and certainly funded some of our work, as well and then when we started to get additional data, then the NIH, I think, realized the importance of this and began to fund it. Although, again, and I think the NIH is right about this, it's not their place really to fund, clinical trials at large scale. It's their place to fund the innovative research that will lead to those trials. And fortunately for all of us, the biotechnology industry eventually came into this, that really helped move all of this much more quickly. But of course, these CARs, we had to figure out what the targets were, and almost everybody focused in on CD19 because it's present on acute lymphoblastic leukemia, non-Hodgkin's lymphoma, and chronic lymphocytic leukemia. So it seemed a target that we could actually go after a number of different cancers, many of which, after they fail frontline therapy, are incurable. And so that provided a place to test the treatments, a target, and we were all sort of ready to go.
Since when do we know that CD19 is expressed on ALL and non-Hodgkin's lymphoma?
Oh, for many, many years. So this dates back to, I mentioned early on the interest in hematopoiesis, right? Mm-hmm. So very early on, the International Histocompatibility Workshop not only worked on HLA molecules, but they also worked on molecules called cluster designation molecules. So when you see something that's CD19, it's cluster designation 19. And this came from work, in the 60s, 70s, well, actually the 70s and 80s, after we had monoclonal antibodies, to actually characterize all the different cells that were produced by normal hematopoiesis. And so what you would do is you would immunize mice and then pull out antibodies and then look at what the antibodies saw and eventually characterize the surface molecules. And these were called CD molecules, right? Cluster designation molecules. And so we were able to realize that, B cells were different than T cells. That's how CD3 came along. And so it was really actually a fascinating time. And the internet, it's actually worth going back because they had an annual meeting, and many colleagues in Europe were involved in the International Histocompatibility Workshop. And every year they would publish, the information on the new antibodies and the new CD molecules that had been discovered and characterized. And this was a huge catalyst for research, but it told us that a molecule like CD19 is present on B cells during B cell development. It comes on very early and it stays on. Right through till differentiation to plasma cells. And so, we now were able to say this is a lineage specific target. So it should be safe, right? It's not going to be expressed in other cells, although now, of course, as you may know, there is a little bit of evidence that it might be expressed in some other rare subsets of cells. So we had targets. We had these CARs. They worked very well in preclinical models, and I think the group of Carl June at University of Pennsylvania deserves credit for pushing this quickly into the clinic. They published their first paper, I think, in 2010, the Kalos paper in chronic lymphocytic leukemia, with a few patients demonstrating that this could work. All of us were working on getting it to the clinic at the time, and very shortly after that, we initiated trials at Fred Hutch, and of course, Memorial Sloan Kettering, the Satellane group also as well. These were really exciting times. I mean, when you start putting these cells into patients for the first time, it's. I remember back to my early days when we first put the first CMV specific T cells into patients, into transplant patients, and sitting at the bedside, almost, tachycardic, perspiring a little bit, and very, very nervous that as those cells went in, the patient might suffer some side effects. But fortunately, the CMV cells were tolerated incredibly well. The patient would sort of look at me and wonder why I look so nervous. The CAR T cells did have some toxicities, of course, and I think we'll come to those in a minute. But what was remarkable was the efficacy. We were treating patients that had very large tumor burdens. They had failed everything. Many of them had failed, the leukemia patients had already failed allo transplant. Some of them, the lymphoma patients had failed auto transplant. Refractory to chemotherapy, had, growing tumors. And it was remarkable. I remember one of the first lymphoma patients that we treated, and of course we hospitalized him to treat him, and he had big lymph nodes in his neck. His neck was 3 times its size because of all these lymph nodes. And the cells went in, and each day when his oncologist around, the oncologist called me up and he said, he said something to me today that he said, I just have to tell you. He said, I can feel them getting smaller. It's like they're melting like ice cubes. And that's exactly what happened. They just shrunk away. And within 3 weeks, essentially, his CT scan was normal.
Amazing.
So when you see that kind of efficacy, it's really remarkable. Now, of course, there were challenges. I mean, we had to figure out the manufacturing approaches for these cells. Again, fortunately, we had done some early work in the 1990s. It was actually my first patent was on a method for growing T cells using anti-CD3 and anti-CD28 antibodies, and actually growing them and genetically modifying them. And everybody sort of used a variety, a variation of that method. And fortunately, companies like Miltenyi and in Cologne and now Thermo Fisher, but at that time, Dynel made beads that had the antibodies that we could actually use to activate and expand the cells. And again, I think there's still work going on in terms of evaluating, the optimal culture methods. And I think it's an area that I think maybe we can talk a little bit about in future research, because I think that the qualities of cells is ultimately going to be very important in terms of reducing doses and getting maximal efficacy. But anyway, so we found, we had high efficacy, as all the groups did, in acute lymphoblastic leukemia, non-Hodgkin's lymphoma, and in chronic lymphocytic leukemia. And, we also learned there were side effects. And this is, again, an area of real intense study now, the cytokine release syndrome. T cells, when they're activated, normally produce cytokines, molecules that activate broadly our immune system. Of course, when we get the flu, we all have a fever, we ache, we feel terrible. Those are cytokines that are causing those effects. And when you put in T cells that get highly activated and undergo proliferation, this is a living therapy. So you can put in a small dose of cells, and these cells will massively proliferate in the patient, just like our cells do when we are recognizing a virus and we're clearing the virus. They do the same thing when CAR T cells clear a tumor. But producing these cytokines can cause problems if there's too much tumor and the T cells, continually have to stay activated for several days. And so this turned out to be something we had to learn to manage. We're still understanding the complexity of the cytokine networks that are activated, because when T cells produce cytokines, they act on other immune cells, and those cells produce cytokines. And so it becomes this orchestration that we've initiated, and we have to, figure out what the best way of managing it. I think we manage it We target one cytokine, interleukin 6, that's produced mostly by the myeloid cells, not so much by the T cells. They're activated by the interferon that's produced by the T cells, and we can target that with specific antibodies either to the cytokine or to the receptor. But in general, what we use is steroids, dexamethasone, to dampen cytokine production and try and control that part of the response. I think we've learned to manage that fairly well. In the early days, it was pretty frightening because there was this reservation about intervening because we didn't want to stop the anti-tumor activity. And I think we've sort of gradually learned how to manage that. And that's where, for young trainees, it's important to understand the scientific underpinnings of the therapies you're giving. It's also important to be a really good clinician. Because we benefited by the clinical acumen, I call it, of a clinician who said, we have to do something here. If we wait any longer, we're going to have a real problem. And that's the advantage, I think, of physician scientists, is they can actually give you both of those perspectives. And then an unusual toxicity that nobody really expected, called immune cell effector neurotoxicity, this ICANS. This was unexpected, and in some patients was incredibly severe. In fact, some patients actually died of neurological complications. Again, I think we've learned that those toxicities, both CRS and ICANS, are in part dose related. So if you give too many cells to a patient that has a very high tumor burden, in part tumor burden related, that you're at higher risk for it. And so again, we've learned to manage that either by reducing the tumor burden beforehand, or reducing the dose, or intervening early in patients at really high risk. So we've learned how to manage those, although they still can be problematic. So at our center at Fred Hutch, we did a very large study that was led by a former postdoc in my lab, Cameron Turtle, where we really tried to put patients on the trial in cohorts. So we had a cohort of acute lymphoblastic leukemia, we had a cohort of non-Hodgkin's lymphoma, we had a cohort of CLL. And we treated them the same way, with the same cell dose escalation to figure out whether those tumors should be treated with different doses. We had used different lymphodepletion regimens, and I'll come back to why that's necessary, to really try and understand the principles of using cell-based therapies. So I should mention lymphodepletion, because it's not just enough to put these cells in. We actually condition the patient by removing the endogenous lymphocytes using chemotherapy. And the reason for that is that the endogenous lymphocytes utilize what we call homeostatic cytokines, molecules like interleukin 15 and interleukin 7, to maintain their survival. And by reducing those cells numerically, when our cells go in, the adoptively transferred cells, they now have access to these cytokines to promote their survival and their initial proliferation. And it turns out that's very important in optimal engraftment of the infused cells, and also in their initial expansion and survival. But the intensity of that lymphodepletion, of course, if you're too intense, you can cause side effects from the treatment. And if you're not intense enough, you don't get effective lymphodepletion. So we worked through a lot of that, and I think established principles that now are sort of generally used in the field. So I mean, this is exciting. There, the biotech, was obviously very interested in this. It was a new therapy. It was highly effective in these blood cancers, and several companies took over these approaches, and are now, two are approved, and we're hoping that the Bristol Myers, the Juno product, will get approved soon as well, at least for lymphoma and acute leukemia. Now, there's a lot of work.
What were the biggest differences between the three groups?
There's really not. So the difference is that the Kite product that's now been taken over by Gilead, that uses a CD28 zeta signaling domain and a CD19 scFv. The Novartis product and the Juno, now BMS product, uses a 4-1BB CD3 zeta. And if you look overall at the data, there's not so much of a difference in efficacy. there may be differences in the behavior of the cells. Maybe not surprising given that the signaling domains are quite different, 4-1BB versus 28. The CD28 zeta cells tend to proliferate very rapidly and don't persist as long, and may be associated with more cytokine release syndrome. Although again, I think that's being managed very well with dose and early treatment. The 4-1BB zeta cells tend to persist longer. So there are patients out 2, 3, 4, even 5 years, I think now in some of Carl's patients that still have persisting CAR T cells and B cell aplasia because they, of course, continue to see CD19. And this probably has to do with the signaling of these different receptors. So we published a paper in Science Signaling a year or so ago, where we actually compared the signaling between these two receptors, and it sort of predicted their clinical behavior. The CD28 zeta CAR signals much more intensively and much more quickly than the BB zeta CAR. And that's partly due to the other molecules, the other signaling molecules that associate with the receptor, and the fact that the CD28 zeta CARs tend to have a higher level of tonic signaling. They're signaling a little bit even without engaging their target molecule. So we think that kind of thing can, that kind of work can inform, the future generations of these receptors that would potentially tone down signaling so you don't get the severe CRS, but actually might provide signaling characteristics that would give you superior efficacy. And we continue to work on, completely novel CAR designs that we are hoping will do that. As well as improve the sensitivity to recognizing the antigen, because that's sort of the other problem, is not everybody responds. And we're now starting to understand why. What are the mechanisms of resistance to these new therapies? And that's going to be an area of I think, intense investigation and innovation over the next several years. So one mechanism, of course, is that if you're targeting one antigen, the tumor can lose it. And so the escape of CD19 negative cells was a problem. And it's a little bit different perhaps in the different cancers, but maybe as high as 20 to 35% of patients will escape with a CD19 negative variant of the tumor. So that's a problem. And also even, people have moved on to target additional B lineage molecules like CD20 and CD22. And again, we're seeing the same thing, especially with CD22, is that even there, CD22 low cells can escape. So antigen density is a key issue. And
do we know the timeline of this escape? Do you know in general when that's going to happen? Or
well, it can happen early or it can happen late. I mean, it's it is variable, and it may depend on the heterogeneity that you're dealing with in the beginning. But I just would point out one thing, is that even though these CARs are designed based on the principle of a T cell receptor, that is, we can engage these signaling pathways, the binding affinity of an scFv is very different than a T cell receptor. And actually how the receptor triggers the T cell is also quite different. And so if you look at the sensitivity of a chimeric receptor for seeing its target compared to a T cell receptor for seeing peptide MHC, a T cell receptor can be triggered by as few as 1 to 10 peptide MHC complexes on the target cell. A chimeric receptor typically requires, 1,000 molecules to be efficiently triggered. Now, it can probably sense antigen at somewhat lower than that, but it's still at least an order of magnitude, if a log 10 or 2 log 10 higher than a TCR. So one question is, can we change that? I mean, can we make chimeric receptors that have greater sensitivity for antigen? We've been working on that and have designed new constructs that do have better sensitivity. And it's really surprising that actually tinkering with different components of the receptor that one might not think would change the sensitivity actually does. And then, of course, there are major things that you can do based on understanding of signaling that will I think, help a lot. But one thing I just wanted to say there is that we don't really fully understand why a T cell receptor has that exquisite sensitivity. There's a whole variety of models for why the T cell receptor can do that. that can be, serial triggering. It can bind on and off the peptide MHC that allows it to amplify its signal. There's a mechanoreceptor function that the peptide MHC actually provides a mechanical force to the T cell receptor that can be involved in signaling. There's a theory called kinetic proofreading about how the signal amplifies in the cells. So I think as we start to understand that, we can apply some of those principles to CAR design and improve sensitivity. And I think that's going to be important because, as we move to other cancers, and the major other tumor now that's susceptible to CAR T cells is multiple myeloma, where targeting a molecule, for example, like BCMA, which is another, lineage-specific molecule that's expressed on plasma cells, it's very clear there that you eliminate the antigen high cells most effectively, and antigen lower cells can escape. One approach that we came up with to overcome that's actually now in the clinic already, is that we looked at what regulates the level of BCMA on the surface of the cancer cell and of myeloma cells. And it turns out that BCMA is cleaved by an enzyme called gamma secretase. So on the cell membrane, it's cleaved in the transmembrane domain and it's constantly shed. So this molecule is constantly being released. And indeed, you can find soluble BCMA levels in myeloma patients. And that was sort of one of the keys that, hey, this must be being shed. So, there are inhibitors of the gamma secretase enzyme. These were developed actually early for many years ago for treatment of Alzheimer's disease, because it was thought that gamma secretase was involved. Turned out they didn't work for that, but most of the big pharmaceutical companies had these drugs on their shelf. And so we partnered with one to get one of the gamma secretase inhibitors and did the preclinical work demonstrating we could improve the activity of CAR T cells targeting BCMA. And now that's in the clinic at a trial led by Damian Green at Fred Hutch. So I think this idea of combinations that influence the level of antigen on the cancer cell are going to be important. And I think we have to think even beyond those types of combinations that would enhance the function of T cells as well. So those are sort of the successes, but how do we deal with this heterogeneity of antigen expression? So I think people are now designing receptors that would see more than one molecule, or infusing cells that see CD19 then CD20, or that see both CD19 and CD20 at the same time. And I think that I suspect will help us get closer, but of course it imposes a level of complexity that if you have to make multiple cell products, and these bispecifics receptors don't often work so well, seem to lose binding affinity of one or the other molecule. So I think there's a fair amount of work still to be done there. But the question is, how many antigens do we need to target to really fully eliminate the tumor? So I think there's going to be a lot of work going there. But the reality is that if you look at those blood cancers, leukemia, lymphoma, multiple myeloma, patients are benefiting now from CAR T cell therapy. And I think that's fantastic. And when we have a treatment option for patients, it's not ready to replace conventional therapies, but it's improving both the duration and the quality of life for many patients. Well, I have to tell a little story of one of the first myeloma patients that we treated, who had failed everything. He'd failed 2 autologous transplants. He'd failed all his therapies, and came in, and he was actually very sick, and we almost didn't treat him with CAR T cells because we thought he's too sick. He's at high risk of complications, but we did go ahead and treat him, and a few months later, Damian Green called me up because he had come back for an evaluation. His myeloma was completely gone, and he was surfing in Hawaii. And I think, he had a miserable quality of life for at least a year before we treated him with CAR T cells, and now was suddenly having this wonderful quality of life. So, sometimes extending life by years, even if you're not curing patients, can be very, very important. And I think eventually, there's no reason in my mind, and I sometimes get criticized by my colleagues, but I think we ought to be, looking at CAR T cells and multi-specific targeting and strategies that aim to cure. I don't see any reason why, we shouldn't be really doing that. And I think it's One of the challenges, I think, is with these different companies now driving the clinical translation of this work, it's a little bit hard for academic centers to fund the clinical trials to actually do those kinds of experiments that are aimed at that much higher goal. And I just hope that we eventually continue that work and get there. I would point out that B-cell malignancies are relatively unique, right? Because they have these lineage-specific molecules. So it's very hard to target T-cell leukemias or lymphomas with CAR T-cells. And there are strategies to do that, using gene editing of the CAR T-cells to remove the target on those cells, so they could work against the tumor. But of course, they will eliminate normal T-cells as well, and you'll have to have suicide genes and things like that. So that's a problem. And then acute myeloid leukemia is not yet targetable, and largely, again, because the molecules that it expresses are on other myeloid progenitors or even hematopoietic stem cells. And so you can't effectively target it. So how do we go after these more difficult blood cancers? And so I think there's two approaches that are being taken. One is this idea of gene editing. So Sar Gill at University of Pennsylvania said, well, we can target CD33 if we do a transplant with CD33 gene-edited hematopoietic stem cells, right? So that they will give rise to neutrophils that don't have myeloid progenitors and neutrophils that don't have CD33. And so that's one approach. Pretty complicated, because you got to make a CAR T-cell and a gene-edited stem cell product. The other is approach that we've been quite interested in and been working on with David Baker at the University of Washington, who's a synthetic protein design engineer, arguably, actually not arguably in my mind, but I think maybe in some people's mind, the most advanced synthetic protein design person in the world. And we've been developing what we call logic gates. So that what we can do is, rather than targeting one or a couple of molecules, we can target a constellation of molecules. So the tumor cell would actually have to have 2 or 3 or maybe even 4 molecules to be recognized. And you can impose what we call AND gates, where you'd have to have 2 molecules or 3 molecules for the T cell to be activated, or gates, where to deal with heterogeneity, or what we call NOT gates, where if the normal cell has an antigen that is also shared by the tumor, or not shared by the tumor, but it shares other molecules, you can actually turn the CAR T cell off from seeing the normal cell. And we published the sort of proof of principle of that approach in Science just a few months ago. And Wendell Lim's lab at University of California, San Francisco has also got a different system for logic gating, using where it's cell intrinsic with what he calls these syn-notch receptors. So I think that this is where the molecular engineering, I think, is now taking on a place where we couldn't conceive of that 5 or 10 years ago, to start to deal with the problems that we've seen in the clinical trials and maybe come up with the next generation. I would say the other potentially very disruptive approach to what we're doing now is this idea of in vivo engineering of the T cell. So rather than having to take T cells out by an apheresis and engineer them in the laboratory, grow them up and put them back in, what you might do is target a gene delivery system to the T cells in the patient and introduce the CAR gene, transgene, have it expressed, and have it see the tumor. And of course, that would be very disruptive technology if it could work. And it does work in preclinical models. So, so stay tuned. I suppose it could be disruptive for the field in the future.
Maybe just a naive question, but if you infuse something into the body, how do you monitor it?
Great question.
Do we have instruments that we know? Okay, we measure 1 week after infusion, 2 weeks, 3 weeks, or months later.
Yeah, so it's a great question. Indeed, this whole field of what we call correlative studies, which is to monitor the cells and what's happening in the patient, the cytokines, all of this stuff we monitor. So what we did, so there's a couple of ways of doing it. You can take out a blood sample and you can actually use a recombinant protein that's linked to what we call a fluorophore. So something that you could read out on a fax machine, on a cell sorting machine, that would bind to the chimeric receptor, right? So if you have a CD19 targeting receptor, if you had a CD19 molecule that was linked, for example, to an FC and then linked to a fluorophore, you could actually count the cells. We did something a little bit different than that, is that in our CAR transgene cassette, so we have the lentiviral vector not only encodes the chimeric receptor, but then downstream of a ribosomal skip site, it encodes another cell surface molecule. This was developed in Mike Jensen's lab. It's a truncated epidermal growth factor receptor. So it actually is part of the EGFR molecule. It sits on the T cell, won't bind EGF because we deleted that part, and it won't signal because we deleted the intracellular signaling domain. And of course, we have very good antibodies for EGFR. So you can actually track the T cells that express EGFR because no other T cells express EGFR. So this allows us to very precisely count them and see them in the blood and actually sort them back out to study them. So what is their activation state? Are they becoming exhausted, for example? It's turned out to be a very useful tool to track these cells. And what you see when you do this is when you study patients over time is you put in a very tiny dose of cells that you might not be able to see at all on day one. But by day 5, you have often the majority of the lymphocytes. By day 7, maybe all of the lymphocytes, 80 to 90% of the lymphocytes in the blood are the CAR T cells that you can see. And they will continue to expand and then gradually contract as the tumor goes away. So it really is this war that you can actually observe, that the T cells going up and the tumor cells going down. Yeah. and then the T cells will contract just like they do in a viral infection. They after the antigen is cleared, they lose their stimulation and they contract. And so, this is, I think very important to be able to study this and to understand the in vivo behavior of the cells. Because I would point out there's another area that I think is going to come to the fore here, which is, in addition to engineering the cell to express a receptor that would target the cancer cell, we could also engineer that cell to overcome other barriers, for example, in the tumor microenvironment. So T cells have this propensity. It's a very big problem in the solid tumor field called T cell exhaustion. It's the reason why we give these, anti-PD-1, anti-CTLA-4 checkpoint blockers, right? Is to release T cells from this exhausted state. And so, there may be ways of intrinsically engineering T cells to resist this propensity to becoming exhausted, because it looks like it's a differentiation state. And so maybe we can alter that to maintain the effector function. And indeed, there's some early work that Crystal Mackel's done that actually looks very encouraging in that regard in CAR T cells. And we've been working with Crystal in some sophisticated animal models to actually demonstrate that this can have an effect. So there's additional cell intrinsic engineering that you could do. That I think is also going to play a role in making cells. I think what we're really trying to do, if you will, which is maybe a little bit, I have to be careful about saying this because it may sound, narcissistic or a little bit overambitious, but we're really trying to improve on nature. We're trying to engineer the cell in a way that we control its specificity, that we control its longevity, that we control its differentiation, and we control its function. As we learn more about cell behavior, we have ideas about how we can do this. Now, that imposes risks, I think. If you knock out molecules for a reason, other things may occur. One example, for example, is PD-1, which is one of these immune checkpoints. You'd say, well, we should knock out PD-1, the cells will work better, right? Well, it turns out if you look at the data, if you knock out PD-1 in mouse T cells, you can't form memory. So they may function better early, but then they may not form memory. And then Jörg Ruland in Munich has published some very provocative work in Nature that suggests if you interfere with PD-1 inhibitory signaling, that you may make T cells more prone to becoming malignant. So we have to, I think, be careful about our ambitions in engineering cells as well, and do all of this very, very carefully. But nonetheless, as I say, I think that we have these opportunities to think about these kinds of approaches that would really control cell behavior in a more sophisticated way. I think some for safety. I mean, if we had safety switches, the EGFR molecule, I might add, Dirk Busch in Munich with our group demonstrated that could be used as an off switch, that you could actually eliminate the cells by giving an antibody to EGFR and recover B cells and recover function. So that's one example. There are other cell intrinsic switches that are drug responsive. You give a drug and the cell dies. And those kinds of safety switches, I think they're available. But the more genes you put into cells, the more edits you make. You also can introduce problems. So I think there's much more work to be done in that area. But I think it does offer us the possibility of improving these therapies in the future.
When we come to the present, where is the CAR T cell field now, like indications? Can you outline that? Just where have we arrived for now?
Well, so of course, there's lots of work going on in the clinic, in what we call phase 1 and phase 2 trials at various centers, in the United States and China for sure. Many trials now in Europe as well. But the approved products are only approved for diffuse large B-cell lymphoma and for ALL, and that's with CD19. I anticipate BCMA CAR T cells will get approved very soon for multiple myeloma, and so that's where we are in terms of approved products. So if you're a hematologist at an academic center, that's what you can give your patients that's approved. Now there are hundreds of trials going on around the world. With multi-specific targeting, with additional genetic modifications to cells, with novel targets. And I think that, we'll see where those go. I think the good thing in the field is there's lots of activity. And I think that that's good but I think, we also should step back a little bit and make sure that our innovations are directed at what we've learned. So if we've learned why patients are failing, we ought to try and address those issues. And I think they fall into 3 areas. One is the quality of the T cells. We want to make sure that we give T cells that have the capability of proliferating, differentiating to effector cells, and persisting, right? Those are 3 qualities that I tell everybody when they come to my lab. These are the things that we want our T cells to do. And I think we can do better in that regard. Now, I would say it's probably a minority of patients that fail because of the quality of the product, but some do. It may be as high as 10%. If you count patients that you can't make a product on. So that's one thing. The second thing I think we have to deal with is this issue of, why do cancer cells escape? Is it the level of antigen? Is it antigen loss? And so there we have to come up with strategies. And typically that's going to be targeting more than one molecule in the early stages. There are other ways that cancer cells can escape, though. There was a really wonderful presentation at ASH this year from the Stanford group, demonstrating that many lymphomas have mutations or lose expression of CD58. Now that's the adhesion or the binding molecule for CD2 on T cells. And so it's thought to be a little bit of an adhesion molecule, but it's also obviously a signaling molecule. CD2 is another one of these, quote, co-stimulatory molecules. And indeed, there's some work in terms of where CD2 organizes in the synapse between a T cell and the target cell. And actually what the Stanford group showed was that the function really was co-stimulation through CD2. And so what they're thinking now is designing, and showed that they could do this, design an additional receptor. So you have your chimeric receptor that would target the tumor. You have another chimeric receptor that would actually deliver the CD2 signal to the T cells. So if the tumor lacks CD58, it wouldn't matter. And I think that's sort of a beautiful example of studying the patients that fail, identifying the problem, and then coming up with a strategy to overcome it. And then I think the third area that we don't understand so well yet is the tumor microenvironment. And this appears to be a reason for failure in some lymphomas, that there are these other suppressive cells or metabolic inhibitions in the tumor microenvironment that may allow tumors to escape. My lab's been studying that mostly in the context of solid tumors, where the tumor microenvironment is typically very hostile to T cells. Where they can be hypoxic, it can have a lot of suppressor cells from the myeloid lineage that inhibit CAR T cells. And we've demonstrated some strategies there to overcome some of that. So I think, testing hypotheses based on data that you get from the clinic, the clinical trials that are ongoing, I think is really important. And that will lead us to understand, what do we need to do genetically to the cell? What are the other approaches that we can take? And so I like that, we call it, sort of iterative experimentation, where you do something, you learn from it, and you take it back to the laboratory and come up with the next generation.
Finally, just maybe just 2 points. One question in relation to that, because you said what we need to learn from what we know. Maybe in remembrance of McNamara, I think he was a vice president sometimes, or even Secretary of State.
Secretary of State.
Yeah, Secretary of State, sorry. And he said once this famous citation of the unknown unknowns, what we know what we don't know, etc. And then, what do you think, what is the biggest unknown we know for CAR T? What do we know we don't know?
What do we know that we don't know? Well, I think there are a lot of unknowns. Science is, and medical research is in some ways like peeling the layers of an onion, and you never really know. You can imagine what's underneath that layer, but you never really know. You don't know if there's going to be a fungus spot there or not, right? And I find the wonderful thing about medical research and translational research in particular, and particularly in hematology, is the ability to really study what's happening in your patients. And I think the CAR T cells and cell therapy is just one example where, you can track cells, you can pull them out, you can understand their now we can do RNA sequencing, understand their transcription report, we can look at their epigenetics, we can do so much because of the technologies. But with this massive amount of data, we have to be able to interpret it. And then we have to draw our hypotheses from that data. And I would say the one thing that I've noticed, and everybody in my lab now, sort of hears this mantra all the time, is that, that we're blessed by having this wealth of data that we can collect, but it's up to us to figure out, how do we analyze it and interpret it, and how do we come up with real testable, and prioritize the hypotheses. If you look at a gene expression data set and you got 2,000 over, or let's say 200, genes that are upregulated, how do you pick which one of those 200 to study, right? I mean, so you can put some into pathways, you can do all sorts of things, but ultimately you have to make decisions. And some of that is, hunches, but a lot of times those hunches are based on, on your knowledge and your understanding. And I think that's why it's important for students to read and really, not just the clinical data, but read the science that underlies it, and really try and understand it. Because, you know, a creative young mind will look at something differently than an old guy like me that's sort of, you know, that has all these biases that are built in. So, yeah.
Maybe at last, because you gave the point to that. You said you believe in CAR T or cellular therapy as a curative possibility, because trainees always hear that from professors or who from transplanters who believe in that transplant, that cellular therapy, it's important that we have that. It's a huge improvement, but it's not the cure. What do you think will be the changing moment for CAR T?
Let me just go back a little bit and just preface what I'm going to say with when I first came to Fred Hutch, you know, transplant was, you know, of all the patients we transplanted, we were probably only curing 15 to 20%. And, you know, maybe in some low risk subgroups, we were curing a higher number than that, but we weren't doing very well. And now the cure rates with transplant are, you know, in 60, 70, 80% in some subgroups of patients. So, and that was, I think, partly, you know, because, you know, at least at our center and many centers around the world, you know, that the leaders like Dr. Thomas and others were focused on solving the problems. You know, how do we prevent patients from dying of infections? How do we treat graft versus host or prevent graft versus host disease better? How do we reduce relapse? And so you know, we can improve things. And I think no one should be satisfied with where we are with CAR T cells right now. But there are patients that probably have been cured. I mean, arguably, you know, Emily Whitehead at you know, is many years out now. We have patients that are many years out now that have no detectable tumor and are likely cured. So it can be curative. And I think that's why I say that the focus actually needs to be on understanding when patients fail, why they fail. And you know, one of the difficulties is when you treat a patient, you know, and they go back to their home, which may be 200 or 300 miles or in a different state. And, you know, for us, you know, you often don't get the sample right when you need it, when they relapse, if they relapse, and you're not getting the information that you need to get to say, why did this patient fail? But once we categorize the mechanisms of failure, then we need to come up with strategies to improve it. But I think that the goal needs to be to improve the cure rate. You know, the complete remission rate in diffuse large B-cell lymphoma is probably in the range of, you know, 40 to 45%. Why isn't it 90 is the question that I ask, right? And you know, we have to understand that, and then we have to design and improve our therapies to make it that. And I think that should be our goal. And that's going to require the next generation of young, you know, enthusiastic, bright hematologists to take on that challenge and to work, you know, towards that goal.
We'll do that, definitely. Thank you very much. It was a real pleasure. It was fascinating. You just answered all my questions here I wrote down. So you perfectly set out the whole program.
Fantastic. Well, I'm glad to talk with you. It was great. The questions were spot on, and it was a pleasure to discuss, you know, where we've been and where we're going in this field.
Thank you very much, and hopefully we see each other one time.
Yes, hopefully. Yes, we have a ways to go, I'm afraid, but hopefully we'll be able to see each other in person for sure. Enjoy your evening, I guess.
Yeah, enjoy your day. Thank you very much, and see you. Bye-bye.
Take care. Bye-bye.